Perfect. Thank you so much. So good morning and welcome. I'm Cindy Cronenshield with Cambridge Health Tech Institute and Executive Event Director of the BioIT World Conference and Expo. We're excited to be able to finally bring our event to you this week. We really appreciate your patience and persistence as we all navigate this virtual world. In 2002, when BioIT launched, it launched during a critical time in science. The Human Genome Project was near completion. The era of digital biology was just beginning, and the common driver of all of these efforts was data, and the data deluge that was inundating life scientists in every field. There was a need to start bringing together a community of people to discuss the issues of data generation and application, especially those working at the intersection of IT and life sciences. And in thinking about data generation and application, it's critical to translate effective and efficient methods and techniques to new frontiers in drug discovery and development, biomedical research, and clinical and healthcare initiatives. And this translation is made possible by cutting-edge technologies. Much progress in life sciences has taken place over the years, especially in life science technologies. However, the fundamental goal of our community 19 years later remains the same: to discuss the data issues of generation and application and how to make the most of data and data computing to advance precision medicine and science. The future of precision medicine is driven by the questions that we ask today and then powered by the technologies that help us answer them. So this week brings together thought leaders from around the world, like we would if we were to meet in Boston in person, but this week we're meeting virtually. The platform we are using provides many opportunities for you to interact, network, and connect with your peers, speakers, exhibitors, and sponsors. And we hope that you do make connections this week, that you forge collaborations, that you learn about best practices and see what's really happening in BioIT. And on behalf of the BioIT team, we hope you enjoy your week. It's a great time to be part of this community, and we're so fortunate to have you with us. In today's plenary keynote session, we are discussing important topics about data, data science, and strategic initiatives that are taking place at the NIH that address the COVID pandemic and opioid crisis, for example. Our program will begin with the keynote introduction by Scott Parker with Sinequa. Doctors Gregurick and Baker from the NIH will follow with individual presentations, and then Ari Berman, who is CEO with BioTeam, will moderate an interactive Q&A panel at the end. I want to thank all of our speakers and our sponsor for being here and preparing for today. At this time, I invite Scott with Sinequa to go ahead and share his keynote intro with us. Thank you.
That's great. Thanks, Cindy. As mentioned, I'm Scott Parker, Sinequa's Director of Product Marketing. I'm honored to be here to introduce our keynote speakers. Both speakers serve as directors at the NIH. As you may know, NIH's mission is to seek fundamental knowledge about the nature and behavior of living systems and the application of that knowledge to enhance health, lengthen life, and reduce illness and disability. As an enterprise software vendor, Sinequa's mission is to provide a means for organizations to make explicit knowledge readily available for all employees. So let's review why explicit knowledge is increasingly difficult to apply readily and consistently. Researchers, biostatisticians, scientists, and other knowledge workers, especially at large, globally dispersed healthcare and life science organizations, we're becoming increasingly disconnected from the information, the expertise, and the insights that are critical to success, and at the same time, they're hidden within this increasingly complex and expanding digital landscape. And it's really caused by several factors, as you can see here. The content increasingly sprawls across many repositories and often multiple clouds. The majority of the content, so-called unstructured data, is routinely excluded because of its inherent messiness. And the people and the groups responsible for generating the content and the data, they're highly distributed, and this leads to data dispersion. So with these challenges in mind, Sinequa developed an intelligent search platform that brings related information together from across storage silos and across geographic locations. This information is automatically enriched to make it more findable and more discoverable and ultimately more useful. And the result of this automation manifests itself in different ways. In the most basic of scenarios, intelligent search frees content and data from these so-called storage silos, and it transforms it into relevant information and often actionable insights. It enables unified views of key topics, such as patients or drugs. And it also means the ability to identify sensitive information that is likely lurking in the data and is a potential liability. And without exception, it improves individual and collective productivity, which allows development organizations to go to market faster. So our platform serves as a foundation for these intelligent search applications. Some of them are shown here that can address a pretty wide range of use cases. Each of these intelligent search applications securely leverages the same index. That's part of the magic or the secret sauce. What this does is minimize the footprint of the index and maximize its efficiency, because the same content can be leveraged securely in different applications. And if you translate this into economic terms, it means that for every project deployed on the platform, the cost per document goes down while the value per document increases. And that sometimes happens exponentially, depending on the projects. So, why Sinequa? Well, we provide a complete end-to-end solution. It's been tested and proven by some of your organizations, some of the world's largest organizations in some of the most sophisticated environments. And according to the major analyst firms, Sinequa has been declared a leader in our market. We've been a leader for the latter half of the last decade reliably. We've seen other vendors come and go from this leadership status, but Sinequa's remained. So that's a little bit about Sinequa. Again, we're pleased to be back to BioIT, obviously virtually this year. We'd like to welcome you. And without further ado, I'd like to bring up our excellent keynote speakers. I'm going to stop sharing.
Thank you so much, Scott. I'm going to start sharing. It'll just take me one moment to get everything set up. So just bear with me while I switch the display and also switch the monitor. I am super excited to be at BioIT World today, and I sure wish we could be together in Boston. However, the agenda looks fantastic, and I'm really looking forward to our virtual meeting. Just hoping that this is going to work. Someone's going to tell me if it's not. So I think we're making real headway to create data that is fair, findable, accessible, interoperable, and reusable. And sometimes it feels like when we get there, we run a race. Yes, our data is fair. The end. Or is it? Really, sometimes when I think about making data shareable, it's really just the beginning of a lot of really interesting and great science. And my talk focuses on getting to that starting point and maybe just a little beyond. When I think about where we are at NIH and other agencies, I think we've already started the race, and I think we're making a lot of great progress. So I'm going to tell you a little bit about some of the things that we're doing at NIH, and then I'm going to hand the ball over to my excellent colleague and friend, Dr. Rebecca Baker, to tell you about HEAL. NIH supports research in many different areas, from biophysics to structural biology. You're seeing a cryo-EM structure up there on your upper left, to neuroimaging, for example, of the brain, to cellular dynamics.
Susan, yes. Sorry, it's Scott again, but your slides, they don't seem to be advancing.
Okay, hang on. I'm not sure what is the problem.
Maybe try sharing and resharing again. That worked last time.
Okay, so stopping and resharing. I am now not on my VPN, so let's hope that works.
There we go.
Okay, so if this doesn't work, I'm going to have to do something a little bit more old-fashioned. All right, so this is fair. This is the image of we're just starting the race, and we're off to a really great start. And you will tell me if you have now seen images of different types of data.
It is working now, yes.
Thank you. Okay. We do support a lot of different science at NIH: structural biology, neuroimaging, cellular dynamics, tissue engineering, and even biobehavioral studies. And these are just only a few of the really exciting areas that we're supporting. And when we think about tackling data science at NIH, sometimes for me as a biophysicist, it's a little overwhelming. I'm hoping that you're seeing a nice painting of a ship. It feels like there's data everywhere and I'm swimming in a big ocean of data. And it can be a little bit like feeling like we're lost at sea. And getting a handle on this data is really a grand challenge. So tackling data, it's just the first step, but it's an important first step. When I was as an undergrad and a grad student, I used to go to the library, to the stacks, and I would spend an afternoon reading papers. And it was really my favorite thing because I'm a closet librarian. Some people know that. And I think it would be wonderful, and it's actually happening today, where journals could link the data that's in these publications. I'm just showing one here of the retinal ganglial cells, linking that data to the journals. And this is an example that came from an NIH pilot that we ran earlier this year with Figshare. And here you're seeing a particular image of these, of the human brain and looking at these ganglion cells. And wouldn't it be wonderful if I could search on this data such that the metadata were computable and I could find this data and others as well? Wouldn't it be wonderful if I could pull that data and any of the analysis tools that were used to analyze that data into some of my favorite platforms like Galaxy or Jupyter or Bioconductor, or just even having the software linked to GitHub that analyze that data? I think the amount of science that we could enable by really rethinking the way that publications are from static to dynamic would really change scientific inquiry and make concepts real of fair data and reproducibility. I love this slide. I really like to think about metadata as a love letter to our future selves, our future researchers. Thinking about how we can make data much more organized by really sophisticated metadata, making it the most useful data that we can, is really a love letter to our future, and our future will thank us for it. Moving on from the concepts of FAIR, what has NIH in particular done with some of the supported data that we have generated? I'm just going to show you one particular example. And this is really putting to life some of the concepts that Lincoln Stein proposed probably 2 decades ago, which is putting our data, our genomics data in particular, on the cloud. And this is enabled through our STRIDES program. There are many good reasons to do this. For example, large-scale analytics is feasible in these type of architectures. It's also possible with HPC, and it's happening in the HPC community. But the biomedical access to the data through cloud computing and through our STRIDES partnerships is unprecedented. And in this particular case, we've moved almost 37 petabytes of public and controlled access data to Google Cloud Service providers and to AWS. Why would we do that? Well, there's a lot of really good reasons why this is beneficial to NIH and to the community. For example, we have faster access to very large data sets. We can easily share data from central or from federated solutions. The availability to compute on data at this scale is unprecedented. And finally, it just makes the whole concept of reproducible science that much more achievable. This is just one particular example, and it's really a fun example. It's from the Ceratus consortium, and this is a partnership with AWS. And what they were able to do was to pull all of the SARS-CoV-2 genomic sequences that have been sequenced over the last 10 or so years from around the world, pull them together into their platform, and then reconfigure the analysis, the all against all comparison analysis for these genomic sequences. They were able to do this in 3 to 4 days by some really sophisticated software re-engineering, instead of the 12+ months that it would have taken to do this exact same research question. And this is really pretty incredible, and it's made possible by putting all of this data from SRA into the cloud, and then to enable partnerships with industry or academics to enable much more sophisticated software engineering. And that gets to the next point. You know, I love this concept that if data is the new oil, then, and this is to paraphrase from Glenn van der Linden, that if data is the new oil, then AI is the new refinery and the clouds are the pipelines. Okay, so just to rephrase slightly from Glenn, if data is the new oil, then software is the new refinery and the cloud is the pipeline. I think that's kind of a cool image. And here's what we're doing to really enable sophisticated software in the cloud. We've supplemented researchers who are enabling software to forge new collaborations between biomedical researchers and software engineers to really enhance their software, their tools, their methodologies with partnerships in software engineering and computer science to make those research tools cloud ready. And I think that this is something that we will continue in the future. Software sustainability, software enhancements, these are all really important and key concepts for making data fair and enabling a biomedical data ecosystem, and it's something that's important to support. So what have we supported over this past year? A number of really great applications in fast healthcare interoperable resources for making clinical data much more usable. A number of cloud commons, including FaceBase, which is a pretty cool facial recognition platform. Biomedical simulations, including new ways to forge force fields and dynamics in the cloud. Biophysics simulations, including blood flow simulations. Genomics software like the one that I've just shown, imaging and neuroimaging software. So there's been a lot of really great collaboration amongst NIH and our institutes, and I have to say that all of our NIH institutes and centers do participate in this program. So that's making software more sustainable. But what about new ideas in data science and artificial intelligence? I'm super happy to say that NIH is supporting and participating in a program that is a partnership with NSF to accelerate innovations in computer science and information science and engineering. And this is building on an existing program that has been in existence for a number of years, the Smart and Connected Health program, but to forge new areas, including areas in information infrastructure. We're looking for applications that develop tools for interoperability and distributed and federated and scalable digital infrastructures. We're looking for transformative data science, unstructured data interpretation, and knowledge representations and visualizations. We're looking for new and design federated and fabricated multi-dimensional sensor systems, and new approaches to support individuals to effectively participate in their own health. Ways to think about automating health, the human in the loop systems, and finally, medical device interpretations. Please look for these announcements through NSF and through NIH in the later part of this fall. I anticipate that applications will be due in the early winter. Let's think about the future. When we think about developing all of this great data science, I think we have to create science that's for everybody. And so we have a number of programs at NIH. The NIH Data and Technology Advancement Data National Service Scholars Program. We are bringing our first cohort of scholars to NIH this fall and winter to work on a number of exciting areas, including neuroscience, unraveling Alzheimer's and disease genomics, supporting cancer, knowledge extraction, new ideas in medical imaging, and integrating cloud-based platforms for genomic research, to name just a few. We will, of course, be participating and offering these scholars programs in the 2022. So I hope that if you're an industry expert or an academic expert in computer science, in information science, data science, artificial intelligence, or other interesting areas, that you will consider coming to NIH for a 1 or 2 year sabbatical program. We also partner with the Civic Digital Fellowship to offer Coding It Forward interns. These are computer science, information science, and sometimes interesting other sciences like economics who come to NIH for a 10-week summer program. This past summer, we hosted 16 students, and this was not easy because the entire program was run virtually. So all of the students did their work completely remotely from their home. We will be bringing 24 students, again virtual, starting this fall to work in 14 of our institutes and centers and offices, and we will continue the partnership. So if you have an undergraduate student or you are an undergraduate student, I hope that you will apply to the Coding It Forward program and perhaps even select NIH and biomedical research as one of your internships. If there's time, I would love to tell you a little bit about the work that we're doing in COVID-19. This is an unprecedented time, and I think that it's more or less all hands on deck to solve this problem. We are generating an enormous amount of data for COVID-19, and we want to put this data in platforms so that the data will be used by researchers. There are a number of great use cases for researchers with this data that are understanding transmission and infectivity, evaluating treatments and interventions, predicting long-term sequelae, making the social determinants of health with exposure data, examining the impact of child and maternal health, and finally, there are a number of technical challenges that we will have to face. In particular, with long-term sequelae, we asked researchers, what are some of the most pressing questions that you would like to face? And what researchers told us is that patients with severe COVID-19, what are their long-term complications with respect to the prevalence and risk of factors for long-term morbidity following COVID-19 infections? And finally, are there any geographic differences between patients and their COVID-19 outcomes? So we have a number of NIH-supported clinical platforms that will be housing COVID-19 data, including Biodata Catalyst for our PEDAL and our ORCID clinical trials. All of us who are enrolling new participants who are COVID positive or COVID tested. The new National COVID Cohort Collaborative, which is looking at near to real-time clinical data from our CTSAs, our CTSRs, and partners. The Alzheimer's Disease Sequence Project has a number of participants who are COVID positive. And finally, a new partnership with societies and the National Institute of Biomedical Engineering and Imaging, the Medical Imaging and Data Resource Center. All of these platforms are housing COVID-19 data, and we want to get this data out to researchers. So thinking about how we can actually get data to researchers is really a question in creating connected data across platforms. And there are a number of things that we can do to enable this. And I'm just going to hit on a few because some of these activities are supported by our office, the Office of Data Science Strategy. And in particular, when thinking about connecting platforms, there are only just a few things that we need to do to enable this. And the first is to create a system where researchers can log in once and find data across. It seems trivial, it's not. This is what the system would have looked like earlier this year. You want to log into different platforms, you have to log in many times, you have to access data across these platforms. It can take a bit of work. It's non-trivial. This summer, with the partnership of a number of our institutes and centers, we've launched the Researcher Authentication Service, RAS. It's the ability for researchers to log in once, and through the work with J4GH and others, creating a passport for our researchers and a visa for the data so that researchers are able to find the data that they're interested in working with and that they have already been authorized to do so. So in a sense, it's quite a bit faster. It's a first step, and it's an important first step. This year, we're also piloting a system to link data across our platforms. A way to think about data that could be in multiple sites, for example, N3C, Biodata Catalyst, or all of us, or even additional sites, such as your senior living system if you are an older adult, or your healthcare system for your EHR records. Tokenizing your participants and your data across these systems and then linking these through an honest broker is one way to de-identify and link data across systems. And so this is not new. It's certainly just something that we will be piloting across a number of our systems to really make deduplication and linkage of data across a federated system a much more feasible endeavor. There are another activities that we could think about and work on, including data discovery across platforms. And there's some really interesting work coming out of GA4GH with FASP, and a shout out to Ian Four, who's working on this at NIH, and Picture coming out of Harvard. There's other work that we can do in enabling workbenches and tools across platforms, linking these with Jupyter, GitHub, and other resources. Most difficult is the dynamical and always fluctuating policy with data access. Each system has its own way of enabling data access, and so policy resources for data access is important. And finally, information systems that allow you to navigate not just data, but information across systems. All of these make interoperability across clinical data platforms a reality for the future. And with that, I am going to pass the ball to my colleague, Dr. Rebecca Baker. I think we've made progress. We have a long ways to go, but to me, the future looks bright. And I thank you very much for the time together. I look forward to your questions.
Thank you, Susan. Let me try to now share my slides. Okay. Can folks see the slides in front of you?
Yeah, we can see it in the slide mode.
Okay. Great. So thank you, Susan, for the introduction. Susan is a terrific colleague, and what I'll tell you a little bit about is how through the NIH HEAL initiative, or helping to end addiction long-term, NIH is trying to seize on some of these data advances and use them to provide scientific solutions to a really serious problem facing our country, and that is the problem of opioid misuse, addiction, and overdose. So here's the crisis that we face. Hopefully I've advanced slides and you can all see the next slide.
Yep, seems to be working fine. Thanks.
Thank you, Ari. So the national crisis of opioid misuse, addiction, and overdose is devastating communities across our country. You can see here that there has been a dramatic increase in communities nationwide in overdose deaths. The majority of these deaths are associated with an opioid drug. And this dramatic increase resulted in the declaration of a national public health emergency in 2017. 2018 is the last year that we have great data from, but what we found is that in 2019, these numbers continued to go up because this public health emergency is rapidly evolving, and because of the rise of the deadly synthetic opioids such as fentanyl. So over time, the crisis itself has evolved, and what started with perhaps an over-reliance or over-prescription of prescription opioid medications and people becoming dependent on those medications resulted in a rise of heroin use and overdoses associated with heroin, which is more powerful than many prescription drugs. And then ultimately, in this third phase, a significant and really dramatic increase in overdose deaths associated with the very powerful synthetic drugs, synthetic opioids, fentanyl and carfentanil. So these data are provisional, but they show that despite the really hard work of folks on the ground and in the community and policy changes in government, we still haven't been able to make a big dent in the overdose deaths across the country. And then what's worse is that then COVID came and overdoses continue to increase and increase dramatically. So these are provisional data, but in a number of communities across the country, we've seen signs of overdose increases almost to 50% compared to 2019, year over year. And a lot of confusion about where these overdoses are coming from, who is most at risk, because during this time, we've also seen a dramatic increase in drug use overall. So in a recent report from Millennium Health, we found that, for instance, fentanyl use in the time between July and when the COVID pandemic national emergency was announced, fentanyl use went up by 67%. So there's a really dramatic crisis that we face related to the use and misuse and overdose of these deadly opioid drugs. And the research community has been trying to keep up, trying to provide scientific solutions, trying to understand what this community faces. And as researchers, we recognize that we would not be able to address this crisis without addressing the millions of Americans who experience pain. So 50 million Americans experience chronic pain. About half of those experience severe daily pain. And 20 million experience pain that impacts their life so significantly that they can't go to work or carry out family responsibilities that are really meaningful to them. So in light of these two crises, the crisis of opioid misuse and addiction and the crisis of untreated, unmanaged pain, the NIH came up with our research response, which is the NIH HEAL initiative. We were fortunate enough to receive from the Congress significant investment, and we really trained over 25 different research programs that span from prevention research, basic science, understanding pain, understanding new targets for pain, a large number of clinicians for what types of people, and then significant right now and put it into place in real world settings. And recognizing that because of the urgency of this crisis, we really needed to work hand in partnership with communities, sector, and our federal colleagues, we've, through the initiative, established a large number of collaborations and partnerships, as well as collaborations and partnerships across disciplines, seeking to leverage all of the expertise we have, all the knowledge and evidence to provide scientific solutions to the public health crisis of opioid overdose deaths. So I'll now tell you a little bit how our initiative is organized. You'll see we have two primary goals. First is both research and science improvement and prevention for opioid misuse and addiction. So this comes up with novel medicine treatments for opioid use on newborns and children as they grow older, really coming up with novel prevention and treatment strategies that take into account the whole person, sleep, mental health, etc. And then once we have this knowledge, rapidly converting it into clinical guidelines and real world settings so people can take it on and make a big change. And I would point out here at this communities have not always, communities of researchers, these communities of clinicians, communities and care have not always connected one another. And what we found is that there were certain groups, there's fragmentation within the research community. People will focus on one disease condition that's associated with pain, or they will focus on one treatment approach for opioid use disorder, not feeling necessarily connected with the other factors playing into the patients that they serve. However, when we launched the initiative, we found that many of these researchers were really eager to share this mission, this important mission of stopping overdose deaths with their colleagues in these other fields and scientific disciplines. And I think, as I'll tell you about today, our strategy towards data will play a really big part in enabling these researchers to take on more, to be more aggressive in the research questions and take a wholer look in the research questions that they pose, and accelerate our progress towards treatments and better outcomes for patients. So I'll now tell you a little bit about how in some of these research focus areas we've tried to provide scientific solutions, laying out some of the public health problems that we seek to address. So the first is that many people with opioid use disorder, the clinical term for opioid addiction, do not receive appropriate treatment for their condition. So there are 3 FDA-approved treatments for opioid use disorder: methadone, buprenorphine, and long-term naltrexone. Many people never access them. And so through our HEAL research efforts, we're working to test the integration of these interventions, these evidence-based interventions, into community settings. And so that can be justice settings where people come through. Frequently, people with opioid addiction come through the justice community, or emergency room settings where people come when they're really seeking help, as well as a research study, the Healing Communities study, that seeks to pull together all of these interventions and all of these evidence-based approaches to prevent opioid overdose deaths in communities in 4 states that have been hit really hard by the opioid crisis. Our next problem that we seek to address is that even when patients are provided medication for the treatment of opioid use disorder, many of them don't stay on treatment long enough to achieve long-term recovery. And I think in the past, there's been a sense that people go off treatment because their lives are complicated, and that's true. But we also want to help them by providing medications that work better for them, that are more user-friendly, longer lasting, and innovative. So HEAL Research is working to develop immunotherapies that could be given to certain high-risk patients to prevent relapse and overdose. Extended release formulations, so instead of going to a doctor every single week or taking a medication every single day, you could take one once a month or every couple months. And then lastly, medications to address craving and other parts of the cycle of opioid misuse and addiction, not just treating the severe aspects of opioid use disorder. So this research builds on a lot of work that NIH has done in the past, really seeking to understand the patient's experience and help them through these clinical tools accomplish long-term recovery. Another problem that we face is that not all pain is the same, and not all patients with pain experience pain the same way. There are biopsychosocial aspects, but then there are also aspects specific to different types of pain: nerve pain, musculoskeletal pain, pain associated with specific health conditions. And we're working through the initiative to really find ways to study and describe that so that our research efforts to come up with new treatments can be most effective. So that means coming up with a biomarker for pain. If you've ever been in hospital and someone's asked you how much pain you're experiencing, a lot of times they'll ask you to rate your pain 1 to 10. We're trying to do better than that and come up with objective measures as well as endpoints and signatures that can be used in clinical trials. And we're also working to test non-addictive treatments that are coming through the pipeline for specific pain conditions, recognizing that one treatment might work better for one condition than for another. A huge challenge for the clinical community is balancing pain control with the risk of opioid therapies. So opioids can be very effective for treating pain in the short term, but they often don't work for chronic pain. And in the long term, they present significant risks of misuse, overdose, and other side effects of opioids. So what we're working to do through the HEAL initiative is to test new non-addictive medications and other approaches for treating pain through new phase 2 clinical trials networks. We're partnering with the Center for Medicare and Medicaid Services, CMS, to test acupuncture for the management of chronic low back pain in older adults, and really work to understand how the non-pharmacological pain management strategies, such as acupuncture, such as cognitive behavioral therapy, or other complementary and integrative approaches could be put into place in the healthcare setting. So it would be easy for clinicians to tell their patients, this might work for you, and then put it into practice. Another problem that HEAL is taking on is understanding the challenges related to caring for infants born exposed to opioids. So this is a devastating consequence of the opioids crisis, that infants are born every day. They were exposed to opioids in utero, and they come out and they undergo withdrawal, or neonatal opioid withdrawal syndrome. And hospitals and NICUs don't know how to treat these babies. There's not an established standard of care. And so HEAL is working to understand how to best assess these infants when they're born, looking functionally at whether they're able to eat, whether they're able to sleep, and then also understanding the best pharmacological management, what treatments work best for managing withdrawal and getting them out of the hospital safely and soundly. Lastly, HEAL research will address the long-term consequences of these early opioid exposures in the context of all the other environmental exposures that babies from these families might face, and really to take a scientific look and track the effects of these prenatal exposures on brain growth and cognitive development. So as you can tell, we have quite a lot of different aspects of the HEAL initiative. As the crisis is so broad and cross-cutting, we have themes, cross-cutting themes in our initiative, and that includes really working to make sure that we engage research participants as partners, work with our patients and other stakeholders, so that when we design the studies that the initiative is supporting, we're making sure that the science and the evidence that these studies produce is meaningful to our patient communities. We're also taking a progressive approach towards the sharing of data coming out of the initiative, making sure that when these studies publish their findings, informed or interested members of the public can read about them and put them into place right away. So for that, we have a public access and data sharing policy that requires that when HEAL investigators publish their findings, their primary data is immediately available and accessible to the public, and we're putting together a data infrastructure to support that. And then we're also working with our teams of investigators to harmonize our measures and make sure that when these primary data are made available, people can carry out their own synthetic cohort studies or pose their own scientific questions and come up with their own scientific solutions to the opioids crisis. So as you might imagine, with so many different types of research, there are many different types of data that the HEAL community of investigators is expecting to generate. So lots of imaging data, lots of demographics data, genetic, behavior, lots of different data types will be generated. And some of our studies are working really proactively to harmonize the common data elements or elements of the data collection in the initiative that will help us facilitate comparisons between studies, help secondary researchers access quality and meaningful data, and allow really quick comparisons of results from these different studies. So if one study found one finding and another found something different, how can we quantify the impact of the interventions and reconcile differences in our observations? Some of the specific common data elements that HEAL has selected relate to, or first we have a core set of common data elements that are just questionnaires and domains for the data that are collected so that when it's all deposited and all the research has gotten off to a strong start, people can go in and actually pull data from different studies. But then we also have a large number of additional common data elements that are kind of harmonized measures and questionnaires across our different studies. And the goal here is really to enable research beyond what the initiative itself is funding, but allow the pain community to see this as a place where they can go and find a source of data for their own questions and their own novel studies. An important part of making this real will be, as Susan discussed, making the data fair, findable, accessible, interoperable, and reusable. And that includes the unique identifiers, the systems that make it findable and accessible, the taking advantage of some of the terrific advances that Susan and her team have been putting into place across the NIH. We'll be creative about which of the studies we seek to make interoperable and how to enable that, and really be sure to put into place a structure that allows for the data to be reused and metadata to support the secondary research of the findings coming out of our studies. So in the spirit of imagining what we would be able to do if we had all of these data collected, we had all of these practices in place, HEAL data would be available to the public and could be combined with information such as opioid prescription levels, because opioid prescription is often the beginning of misuse, and connect that with, for instance, a healing community study, how opioid overdoses are affected by changes in opioid prescription practices in specific communities and specific states hard hit by the opioids crisis. Or we could also ask questions that we really don't know the answer to. So which patients prescribed opioid medications are most likely to develop an opioid use disorder? We know that there are hundreds of millions of Americans in pain, and many of them are experiencing overdose, especially now in a time of COVID where so many people are misusing drugs and the overdoses are increasing so rapidly. Who's at greatest risk, and what interventions might be most valuable for those individuals? So as you can tell, we have quite a diverse set of research to support this, this line of inquiry, a data sharing policy and public access policy that requires that the data be made available to the public, as well as the potential to really further open science through our management and sharing policies. we've put together the beginning of an ecosystem through a data platform. I know that this is really just getting started, but our goal is that as our HEAL investigators get to work generating data of these different categories and types, they can be organized in data coordinating centers and data management centers, put onto the cloud, and that the HEAL data platform will allow for these data to be discoverable and therefore accessible to the larger community. And the reason we're working to put together this ecosystem is really to enable a life cycle of data use and reuse and innovation, so that as these researchers carry out these studies, evidence can be shared with the community, new studies can be developed, the data can be reused post for these new questions, but then also communicated outwards to patients and communities, to clinicians, and to our policymaking federal partners and other agencies. But then I would add to that life cycle that we'll also be looking for innovation in new ways. And so our hope is that this initiative will continue to provide material that can be used to pose new questions and combined with new data sets and provide a kind of a virtual platform for new inquiry and the development of new solutions to the opioid crisis, as all of the data science advances that Susan told you about continue to provide opportunities for research. So with that, I'll thank the organizers again for the opportunity to speak to you today, and thank you very much. I guess now I end my slideshow.
Great. Thanks, Rebecca. Thanks, Susan. My name is Ari Berman. I'm CEO of BioTeam, and I'm going to take us now into the Q&A section of this. Rebecca, you might want to stop sharing your screen.
Yes.
Yeah, thank you both for your typically compelling talks. There's a lot going on. And also I want to thank Scott for his presentation on the Sinequa platform. If folks want to ask questions in the Pathable app, please feel free to do it in the chat window there, and we will attempt to answer those questions. So I will start off with a couple of questions as people are gathering their thoughts. So Susan, to you, what is the hardest problem that you think we have to tackle today in data science and biomedical research, and how is NIH working on this problem? I have a really easy question for you. So.
Yeah, wow, there's so many hard problems and so many compelling problems. So maybe I can layer this, and this is just the perspective of what we're dealing with today. I started to talk about the sequence read archive SRA. It's probably now 37 plus petabytes shared between Google and AWS. We can partition that down into small chunks, but imagine that you wanted to have an algorithm that would search across 37 petabytes of data. That doesn't exist. In a couple years, I think that will. But that's just the foundation of hard. Now imagine we have 80 petabytes of data shared across 5 platforms, all of which have their own initiatives and their own common data elements and their own data frameworks. And now we want to make that data interoperable. That's the real meat of FAIR. That's really hard, just to search across 80 petabytes of data all on the cloud. And thinking about ways to aggregate that data that makes sense to researchers. Now layering on top of that, the data is consented in different ways with different access protocols. And now let's think about the data ecosystem that's also computationally an ecosystem. That's just cloud computing, but there's HPC computing for simulations and integrating in data in the loop. It's even harder. And then now there's sensor data that's analyzed via AI on the sensors, fog and edge computing. We can really make this problem hard, so partitioning it down into solvable bits and working up is a grand challenge, and pulling the community together, another grand challenge. I think it takes an entire community to really make progress in data science, not just for biology, but for weather, for economics. There's a whole world of science that will benefit from the work that we're doing here at BioIT World this week. So I'm excited to roll up my sleeves.
Yeah, a follow-up to that, something that I think about a lot is, so there's a lot of data, right? And the data is in many, many different formats. And you know, Rebecca, I guess I have a similar question for you later, but getting back to Susan here, how do we know which data are valuable and which are not? Because I'm not sure that we have done that well as in biomedical research.
Yeah, that's also a really hard question. As a trained physicist, sometimes the biomedical community looks a little bit like data pack rats. And I get asked a lot, why do you save all this data? Why don't you just rerun the experiment? And here's my answer. Everyone has their own feelings, but the data that we're generating in our research experiments are the tangible product of our work. It provides the knowledge for those publications. It is an instance-in-time snapshot of the science that we are at today. When we publish the data and make it available, that's the way that we can enable the future. However, I think that if you ask many biocurators and folks working in the biodata data management field, they will say that probably a very large fraction of that data is left on the table. And understanding how we can quantifiably measure that and move that to not on the table, but in some platform that people are really using, that is also a challenge, because just measuring and quantifying data use and reuse beyond citing it is tough. But I would say that if you ask me, based on my prior work in other agencies, about 80% of the data that we generate is not really reused in any effective way. And that's really a shame, because experiments are expensive and they really are a time snapshot of where science is today.
Yeah, I agree. And you know, it not being used, I think we don't understand what is actually useful and not from some of that data that's just sitting around taking up energy to store. Rebecca, similar question to you, and there's a couple of questions filing in here, so I'll follow up with that. The same question for you. So you showed the vast diversity of data coming in from the HEAL initiative, and you know, I'm sure there's a lot of different you know, research styles and types, and you know, different programs going on. How do we best collect that and actually make use of it in sort of the near term, given that it's such a crisis at the moment?
Well, you know, for HEAL, because it is a crisis, I think that provides us a sense of urgency. You know, there's I think everyone feels the responsibility that the taxpayers are supporting this research, and it is our job to make it available. I think that for HEAL, the question will be, how can we develop the innovative tools and non-biological research approaches that help connect the data that our researchers are working so hard to generate with all of the other pieces of information that are going on. And that can be public health data, but it could also be innovative data science approaches. And so that's something that we'll be looking forward to kind of put it all out there, make it available in a way that's fair, and then use it as a source of new questions and new lines of inquiry that can be connected with all of the other important research going on to help address the opioids crisis.
Yeah, that is the promise for sure. As sort of a related but follow-up question there, one of our attendees asked, you know, if the data is from different sources, how do we bring them in common methods, you know, with common data elements? And either of you, please chime in on that.
Yeah, I could start and then I'll pass over to Rebecca. And she did such a great job with the common data elements. I love that. That is something that we are working pretty intensively on right now at NIH and have been for quite a while. So developing across systems, what are some of the commonalities that we share in our data elements, data formats? That is certainly a first step. I've heard other folks talk about, making the metadata super rich and standardized in some way that it can also be computable. There are really great advances right now in learning algorithms or graph-based algorithms to link metadata that will enable us to integrate it together. It's still a very challenging problem. I think it's an open area of research.
Great. Rebecca, you have anything to add?
Well, I would say, it's been an interesting experiment with the research community working to develop those common data elements. I think folks often come to the research questions they're posing with a lot of experience and history in those areas. And so they will say, oh no, this question is the best question. And we have to find a way that they can pose that question and use that questionnaire or use that measure, but then also collect this core set that can be researched across our different data sets, across our different studies, and sometimes even in a way that that researcher would have never expected. So they might be an expert in back pain, and the questions about patient-reported outcomes that inform how someone's going to respond to a back pain intervention or our treatment strategy. And another researcher might be wondering, well, which of the patients that have a back pain incident or have some kind of post-surgical pain are most likely to develop an opioid use disorder, and how does the dosage over time change? And if we're not collecting certain core data elements common to multiple studies, you won't be able to pose innovative questions like that. So I think in some ways it's a culture change at NIH to say, yes, your research question is valuable, and that's why we asked you to carry out this research, but add on to it these other capabilities so that the research can be enriched and increased across the board and over time.
Yeah, this is a really hard problem. I don't think anyone's really tackled it well. I think some of the sort of high profile data sets that you listed, Susan, they're trying to with common data elements, but there's not a lot of consensus because of the problem you just laid out there, Rebecca, is that these people really are experts, and it all sort of needs to be thought of in accordance. However, how do you synchronize all of that? So some other questions that have come in. Can you please share more details regarding the data segments that are available, like medical images, gene sequences, chemical interactions for drug discovery?
I don't know if that's to Rebecca or to myself, but I can say that probably all of the above, certainly through Strive. We've put a number of really high profile data sets into the cloud, not just the genomic sequences, but the top med data, the data from GTEx through Anvil. There's a number of wonderful data sets in the Cancer Commons Research Framework, including images, as well as proteomics data. I think that if you're looking for data through NIH and through a number of our platforms, you will be able to find the data that is of interest to you. So I don't think that we're leaving any data types out. Just connecting them is not as easy as it would seem.
Absolutely. Any follow-ups there, Rebecca?
Well, I would say this is an area where HEAL is probably branching off a little bit from what NIH has historically tried to aggregate and research across domains. So the genomic data, the large-scale genomic and proteomic data were the first types to be hosted and put out for this type of research. And now we're expanding a little bit into these social and behavioral type studies, different types of electrophysiology data and different types of data within the initiative. We have some genetic data too, of course, but we have a lot of other data and especially clinical data as well, and imaging data too, that will, be coming available, because I probably skipped this in my talk, but we launched in 2018, made a number of awards in the past year or two. So these researchers are just getting these.
Yeah. And it's going to be, I think, a deluge of very diverse data set, which will be very, very interesting to tackle. Susan, I think you will appreciate this question along with me. Do you have a forecast for HPC teams about the possible amount of data that the HPC teams can prepare for in the next year or two?
That's a great question. I sit on an advisory board for a computing director in a couple agencies, and the things that we're hearing right now is that the architecture of the HPCs are changing to be mostly GPUs, which is good and bad, but the good news is that you can anticipate a lot of data. We're dealing with petabyte scale data. I think that that's probably fairly likely, if not already happening in the HPC world. The really cool thing about HPC that is a little harder for cloud, as far as I can tell, is the way that you can interplay the biodynamical simulations with data in the loop simulations. And that's a really interesting concept, not just for biomedical research, but it's also true in the weather forecasting fields, where here the data, the experimental data, can then drive the parameter prediction that is then fed into the biomedical simulations. And we're starting to see a little bit of that through partnerships with DOE and NIH through the RAS. That's a cancer initiative. RAS is a cancer protein. It's responsible for 30 to 40% of all cancers through its mutation. That dynamic of the interplay of the biomedical course and molecular simulations with the AI-driven parameterization is pretty compelling. There was a wonderful talk and paper, I think it was last year at Supercomputing. So in the HPC world, I would say that driving that convergence of data and analytics is going to be something that is truly unique.
Yeah, it's a huge problem, something that I spent a lot of time thinking about too. So it's going to be very interesting. And I'll postulate that I think, as you alluded to, that HPC engineering in itself is going to change a little bit coming in the near future. I think it has to accommodate the data. All right, so another question that's come in, Rebecca, you got a few in a row here. So at a high level, can you describe what all goes into the harmonization effort for the harmonized measures you described for HEAL?
Well, I'm hoping I'm understanding the question right, but it was a lot of person power, how we carried out the selection and harmonization of some of these measures. So we launched a number of studies at the same time. And so we had to go to them and say, what types of data are you planning to collect, and go through what folks had considered to be their measures. In certain cases, add some. In other cases, ask them to carry out another one in place of what they had planned. I think there's, in some ways there's a lot already there. Like there are lists and lists and lists of CDEs that folks collect. So it is kind of a group effort to determine which are the ones that need to be included as core data elements for HEAL, and then work with the investigators so that they can continue to carry out their studies as designed, while also harmonizing with their colleagues in other research efforts.
Yeah, excellent. I mean, again, this is the problem. We keep dancing around. It is really hard to make this work in the community. Another question for you, Rebecca. Wayne, he asked, what is the measurable impact of the HEAL program so far?
Great question. So there are a few efforts that really are on a fast track. One of the areas that I told you about is the development of new medications for the treatment of opioid use disorder. We have found that to be such an invaluable tool in fighting the opioid crisis, that we have these 3 FDA-approved treatments, but they don't work for everyone, and they don't work for everyone at specific stages in their life. So we put out, I think it's about 40 plus grant programs, and already a number of these, almost 10, have gotten their IND from the FDA. So we have a good set of research programs moving forward specifically to address opioid use disorder and reverse opioid overdose. The other area where I think we've seen a significant effort move into progress pretty fast is in the area of treating and assessing the babies born with neonatal opioid withdrawal syndrome. So this is so fascinating because it was just NICU nurses doing what they thought was best. The babies were born, the NICU is very noisy. There are like all these like blinking things and beeping things going off. And so certain investigators decided to test, well, what happens if instead of kind of medicalizing the opioid withdrawal, the nurses and focus on building connections with the moms. And instead of measuring all the things that are wrong with the babies, measuring the things that they are able to do. And so that study, Eat, Sleep, Console, really seeks to find out whether the baby can eat, whether the baby can sleep, whether the baby can be consoled, and working directly with the parents to establish a relationship there. And so the baby and the mother forming a bonded, trustful relationship. And so that has caught like wildfire, as you might imagine. It seems very intuitive, but it wasn't how the babies were being assessed and treated prior to just a few years ago. So we have been able to leverage a lot of infrastructure that NIH made or has built in the past, including some pediatric research clinical sites in places that often don't get NIH grants. So sites really across the country in 40 plus states where infants are being born exposed to opioids. And so they're already recruiting patients and working with the clinical teams in these neonatal hospitals. And I think that that's another area where we'll see a lot of impact in the community really quickly. For other areas, there's significant signs of progress, and Medicare is covering acupuncture for the treatment of low back pain as part of the collaboration with HEAL in our research in that area. But there are so many other conditions and so many other treatments to test that we're really just getting started.
Thanks. That's excellent answer. It's fascinating. It's and frankly, as sad as it is to say this, it's good to hear about something other than COVID. It's it's as much of a real problem COVID is. I'm going to convolve that question a little bit for Susan, actually, and expand it a little bit to, what is the measurable progress that NIH in general has made in data science in about the same time frame, last couple of years?
I think that, and it may not appear as much progress as to the outside community, but inside NIH, I can feel the palpable changes in the culture of data science, and that every IC director is very engaged in data science. And it's because the staff are engaged at NIH, where there are over 80 people working just on the data science of COVID, meeting weekly, sometimes bi-weekly, sometimes 3 or 4 times a week, just to solve this problem. So the concept of the importance of data science, the importance of getting data to the researchers and integrating it into platforms that do talk to one another, it's a high priority. And that's having a noticeable impact on the external community as well as the internal research community. So I would say that there's been a huge cultural change. And that's thanks in part to Phil Bourne and Vivian Bonazzi, who really instantiated the concept of the FAIR data principles and data commons at NIH. That was just the first building block that we're building and leveraging on. So I would say that I wouldn't underplay the cultural significance of the amount of work that we're doing in data science. And now you see it in the community through funding announcements, but also in collaborative work through HEAL, through other initiatives, through COVID, that really brings data science together with all of the other sciences at NIH.
Yeah, so that is a perfect segue into one of the other questions here, and one that I had written down in advance of this, but one of our folks asked, is good data science hampered primarily by technical challenges or cultural challenges? If both, or both. If cultural, what are some of the biggest opportunities? If technical, what are some of the biggest opportunities? I'll open it up to both of you, whoever wants to start.
I can start, but also it'd be really interesting to hear Rebecca's perspective as a consumer of data science and an initiative. But I think, do you want to start first, Rebecca, and then I can answer?
I mean, I would say both, but I would say the technical challenges are getting addressed, and Susan and colleagues across NIH are making this more feasible. And I think 5 years ago, if we were launching HEAL and I said, well, I want all of the data to be reusable and accessible, it would have seemed just too hard. And now it feels something we should accomplish. And then I think that the culture is giving us an avenue to do that. And so, I mean, I worry a little bit about attribution and finding ways to give credit to investigators who do share their data. And I think that that's a work in progress, to really encourage people, but also to reward them for sharing data and giving them credit for the work that they're doing. But I think that the technical challenges are now feel like something we can manage.
Yeah, I agree. I completely agree. I feel like we can manage the technical challenges. There are technical challenges, but they can be addressed. If you can imagine it, you can make it so. It's the imagining that's really where all the brain power relies. And bringing the culture along with you is another challenge. And it's the constant working with people to enable data science that makes it happen.
Yeah, I think, just to sort of add into this a little bit, we've found that culture is one of the biggest barriers to adoption of anything, and specifically across science and biomedicine. There's such a diversity of very valid viewpoints that how do you bring together these viewpoints into technical systems and cultural systems that allow for broad consumption of the results of those in a way that can be more powerful. I think the biggest challenge here, both culturally and technically, in my opinion, is how do you make it easy? That's not easy, right? How do you make it less hard for researchers to do it this way than the way they're currently doing it? And that's a huge, huge challenge in every aspect. Okay, so I'll try not to interject too much here. I have a lot of ideas on this. All right, so one other question that was asked for you, Rebecca, does your system communicate or harmonize with other data sets, or are you planning to, for instance, the Cancer Research Data Commons or other NIH initiatives?
So it's a great question. I would say it's probably the next set of challenges we take on. Our goal has been to really leverage the advances that projects like the Cancer Research Data Commons have been able to bring forward. And so using those types of platforms and using those types of strategies to make our data that we develop through the initiative shareable in the same way that data through the Cancer Data Commons is shareable. Whether we'll be able to use those technological tools to combine them or research across them will probably be one of our next set of challenges. And I invite those who are taking part in today's discussion to send us good ideas. I think that for some of the technologies, for instance, brain imaging and imaging associated with cancer might have some really cool AI tools that you could use for both. And so I think that there are opportunities there, but we haven't really gotten there yet.
I think there's also, I was just thinking through this, there's probably some major opportunities, and I'm sure you've thought of this, for comorbidities between various disease states and pain and opioid use and things like that, like cancer, for instance. I'm sure there's a lot of crossover there. So it might be very interesting from that perspective as well.
Absolutely. And I think that that's really what we're trying to enable through the data focused approaches, is enabling people to go in with those questions. And sometimes what you hear is like, oh, well, we didn't power our study to be able to ask that question, but if you do allow for research across these different data sets and across these different programs, then you can start to ask these more higher order questions about comorbidities or specific populations within different types of studies.
Yeah, Susan, expanding that now to your rather large viewpoint of things, what challenges are you running into with really creating more of a harmonized ecosystem across these very different data sets, data collections?
I would say that some of our challenges are that data science across NIH and across the different platforms of the different work that each institute is doing is very variable. And so listening and learning and leveraging and understanding where each of our different programs are and meeting them where they are is one of our bigger challenges. There's just such a vast variety of data science at NIH, and the needs of the different communities at NIH could be different. And so it's really a question of listening carefully, learning what is needed, and then leveraging to a greater common good. That is some of the biggest challenges. And you know, it's a small staff that are doing this, so it's a lot of work.
Oh yeah, and you know, as we were just talking about, a huge technical, also cultural problem to overcome. Another question that came in was on data literacy, an important cultural element in getting people funded. Getting a person-funded initiative to the people. So basically, I guess what's your perspective, both of you, on data literacy in as a cultural element and being funded as an initiative to start solving that particular problem?
I'd say that that is a great question. Data literacy, data science literacy is always going to be a priority. So training researchers and training citizen science is something that NIH is good at and will continue to do through our many different mechanisms, including our codathons, which is meeting people where they are to bring the ideas of coding and data science to them, to their problems. That's something that we'll continue to invest in, as well as funding through the ICs, through the different training programs. And now there's new initiatives in data science training, mostly through NIGMS and NLM and other institutes, to make sure that data science is included in the training curriculum and in the, for example, the T32 type of mechanisms. And thanks so much to my partners, Dr. John Lorsch and Dr. Patty Brennan, for really instantiating data science training and data science literacy in their existing programs, because I think embedding data science in all of the work that we do really enhances and brings everybody up.
Great.
Thanks for saying that, Susan. I think it's interesting to really, and this is an exercise in inspiration in some ways. So you have, for instance, in HEAL, you have people who have been peer counselors to people with opioid use disorder, and they've been working in the field in community settings for decades. So you don't want to go to them and say, well, you're not doing it right. You know, you should be doing better sharing your data or seeing it the way we see it, but really to provide them with the opportunities, the collaborations, and the inspiration to be a part of this broader effort.
Yeah, and that sort of coordinated leadership, I think, is really important to help empower the community to work towards this. So there's a couple more questions that have come in, and I've got one to follow up on. The session can go another 10 minutes before the Zoom automatically shuts off. So we'll go just a little bit longer here. What are some of the ways that scientists are changing the way they work? Is it process or technology or both? And that's open to both of you.
I have to say that certainly over the past 7+ months that most of us have been working at home, we have gotten pretty adept at remote working, as well as utilizing new tools and learning new architecture. So I think that scientists are much more easily able to use platforms and tools like Jupyter Notebooks. That seems to be a really common capability that in Galaxy, when I was a grad student, none of that existed. And so talking to a biomedical researcher was a deep dive into what looked like Excel spreadsheet mess. Now there's ability to share workflows, to share computational tools in really meaningful and digestible ways. And I think that that's changed a lot of, at least from what I've seen, biomedical research. And now, because of COVID, we have to become much more facile at computational tools and remote working, because that's where we're living right now.
Yeah. You have anything to add, Rebecca?
Oh, I just totally agree. I think it's an ongoing experiment. You know, for us, that I showed some data about how drug use and overdose is increasing during the COVID pandemic. But another thing that's increasing is the ability for people to take part in certain therapies. You know, so it used to be if you wanted to take buprenorphine and be on behavioral support, you would have to show up to a meeting at 7:30 in the morning before you went to work. Now you can Zoom in, or you could Zoom into a meeting with your therapist. And so I think that all of this is happening at the same time, and you know, people can now take advantage of health support services in different ways, and the researchers are similarly working in different ways to access novel tools and novel technologies to carry out their work.
Yeah, so there's been another follow-up point on that, but I want to bring up one that I wanted to ask in advance of that, and then follow up, and that's probably all the time we will have today. So one of the things I've been thinking about a lot is, you know, data science, you know, was a huge challenge at the beginning of the year, right? But COVID-19 changed the landscape of data and collaborative science drastically. What are each of your impressions of what's changed and how it's affected each of your missions? Start with Susan.
I would say that because the pandemic is so present in all of our lives and it's such a high priority for all of us, that it's kicked into high gear some of the concepts that we were working on behind the scenes, the first being research authentication services. Now it's considered an integral part of moving forward, and that is just the first step. There's the thought about linking data through some kind of encryption method. Right now we're looking at this honest broker method, but all of the ideas and work that we had been thinking about, but on a little bit slower timescale, have ramped up pretty quickly into high gear, and they've become a high priority. And so that's the biggest change I've seen, but also in a wonderful way, the way that the researchers, the community through N3C, through Biodata Catalyst, for all of us and others, are coming together to work and solve this problem is quite meaningful. I think that it might be a change in the dynamic of data science research for the future.
Totally agree. Rebecca, what are your thoughts?
I agree with Susan. I see it as kind of accelerating some of the movement that we had been working to put in place, but now it's so present and it's so clear what we need to be able to do that some of the perhaps more arcane things that would typically get in the way, that's not an acceptable answer anymore. And so, you know, there are all kinds of barriers to how the in-person research can take place in the context of COVID, right? People going into medical settings, but that's given, I think, the community some time to think about how some of the data-focused approaches can go in and solve some of those problems and fill some of those gaps.
Yeah, it's been profound, honestly. I mean, and I don't think we talk about it enough in the community. We're all, you know, doom scrolling on Twitter all the time. So, you know, how do, you know, I would love to share the extraordinarily positive coming together of the global research community that's happened, especially in the realm of data science, to really start solving this problem. It's an amazing story, and hopefully something that's permanent. All right, so final question from the community. It's kind of a long one, so I'll just read it out. A follow-up on this point. Some elements in real world healthcare data, such as demographics, drug prescriptions, conditions, etc., can and should be really be standardized across NIH repositories. If you analyze common data CDMs like OMOP, Sentinel, PCORI, etc., they are very similar and even use the same ontologies. Do you agree that this would be valuable, and could NIH lead this standardization?
I'll take that on. Because of COVID, we have actually been looking at this quite intensely. And so I'd say yes, your answer is yes. And putting out those common data elements that do map across, for example, leveraging OMOP is something that we are actively pursuing with the community and with the capabilities of FHIR to map across these different systems. So yes, absolutely. And that's a great question. Thank you so much for raising that.
Rebecca, anything to add there?
I'll just add the perspective of someone who's not, our research doesn't involve COVID directly, but whenever these advances are made, they can be shared with researchers in other fields. And so as soon as standards are established, or as soon as the technologies come along to bridge these different areas, then we can pick them up and apply them to the problems that we're facing. And so it's just, I love to hear about the progress Susan and her team are making.
Thank you.
Yeah, that's great. So what a way to close this session. I think we covered all the ground. Thank you both very much, and thanks for your insights and for all the work you're doing to really help solve some critical problems that can really improve the quality of life of people across the world. So thank you, everybody.
Thank you, Ari and BioTeam and BioIT World for a wonderful session.
Thank you. Looking forward to a good meeting.
Yeah.
Great. So this is Cindy. I just want to thank you all for preparing for today and for a great discussion, and for our attendees for posting such amazing questions. So hopefully people walked away learning about all of the amazing work that you're doing at the NIH. It's really impressive. So thank you for that. Keynote for today, we do have workshops that are taking place this afternoon. So hopefully those who registered for them can join us, and then we'll see you all back here tomorrow. Thank you, and have a good rest of the day.
Thanks, everybody. Much appreciated.